And our next speaker, Ana Martin-Villalba, who you were here when we were in Basel. That's the last time, and I'm so happy that you're back. So let me just find your slide. Wait, try again.
No? Oh, okay, yes. So I'm also happy to be back here. I'm even happier to be back not in Basel, but in Copenhagen. It's a wonderful city. And I'm going to change gears and not tell you about mitochondria, but rather about brain aging and stem cells. And here. So there's a lot of interest in aging, and it's not only about living longer, but also increasing the quality of life. And as we have heard previously, there's a lot of money and interest, growing interest, from the pharma industry, but also from the non-pharma industry to get into this business. And I also put the slides of all the pharma industry that is here in this wonderful meeting. So, but it's not only about longevity and quality of life. I think if you think of, my group works actually in regeneration and repair. And if you think of repair in the brain, you have to work with an aging brain. So it is also about understanding how a tissue works differently when it ages. And the question is also, it is all bad about aging? I mean, if we think of us aging, I mean, from the small toddler that is very active, and then we think of us being sitting around, we become more sedentary. And aging is also about an energy saving. And aging is also about inflammation. And the question is, when do we stop this process of energy saving? When does it become bad? And how can we get the beneficial effects without getting into the deleterious phase of it? So as I said, my group works on regeneration and brain regeneration, and we all know that over millions of years of evolution, we actually lost the capacity to regenerate the brain. But we all have in our brains these stem cells in the adult brain. And I'm interested in how can we awake these stem cells that we all have in our brains. So the stem cells we are looking at are sitting along the lateral walls of the ventricle, and these are these guys here. And so what happens is that if you activate these cells, they just start differentiating, and then you have newborn neurons that are integrated into the network of the olfactory bulb. And they are very important for olfaction, for recognizing your baby, for recognizing your mate, but also one of the very first things that we lose in aging and in Parkinson's disease mostly is that you lose this capacity of olfactions, and not only upon COVID. And there's other stem cells that are even more important, and the ones you think about aging are the stem cells that are sitting in the hippocampus. And these cells are very important for this short-term memory, you know, like entering home, leaving your keys, and knowing where they are back. 5 minutes later, where did you put your keys? And this is something that you also tend to lose with aging. So the capacity to produce stem cells, we think, or we suspected that is reduced in aging. So what we did a long time ago is just to have a look at these stem cells, to see how heterogeneous are these stem cells, how many of them do we have, how plastic are they in producing neurons. And for this, we did single-cell sequencing at the time where you were very happy if you had like 150 cells to look at. And what we found at that time was that you have a layer of heterogeneity in these stem cells, and this is that they become from being in a quiescent state to being active and to differentiate into neurons. And that there's different metabolic programs involved, like you need to shift from lipid biosynthesis and to oxidative phosphorylation, and you need to activate translation, and that this is very important for getting activation. So the question was, okay, are these young stem cells, the same cells that we found in an old brain. And at that time, it was known that hematopoietic stem cells, they actually increase in number, but they decrease function. If you think of muscle stem cells, they decrease both function and number. And for the intestinal stem cells, it's the same. But there was not much of very controversial results about how neural stem cells or stem cells in the brain fare. And just to sum up what we already published is that when we age, we actually decrease the stem cells we have in our brains, decrease in number, but they are still there. And also the output of these stem cells decreases. And another thing that we found is that while this number and function decreases, interferons and the interferon response in stem cells increases, and mostly because of the environment. So it's not that much the interferon response that it increases in stem cells, but actually the neighborhood affects more these stem cells. And what we also found is that the activation rate and the self-renewal of these stem cells, so the activation rate decreases with age, and the self-renewal, which means, if you think of a stem cell, what is a stem cell? A stem cell is a cell that can renew itself and can differentiate into a more specialized cell. So this self-renewal capacity actually increases a bit with age. So this is what you have. You have a reduction of neural stem cells, you have a reduction of their activation, and you have an increase of your interferon response. So if we have a look at and how did we measure this activation rate, was not only by looking at aging mice, but we actually quantify the number of stem cells across different ages, because what we were interested in is in seeing the cell dynamics of this aging process. And something that you can see here is that actually if the points are the quantities, what we measure, and the red is the mathematical modeling, what it predicts. So if you, while aging, you wouldn't actually change the parameters of activation, what would happen is that you actually end up with no stem cells. So actually this is, I want to point out that that decrease in the rate of activation of your stem cells also has good sides, and this is that you keep them in your brains. And otherwise you would actually lose all of them. So what happens when we age? That nature has given us the ability to become more sedentary as our stem cells do. So the stem cells decrease the rate of activation and increase this probability of self-renewal. So we have more dormant, sleepy stem cells in our old brains. So what happens with them? Can we actually wake them up? And to do this, what we do is a trick. We use Temodal. This is just a chemotherapeutic drug that you normally give to patients that have brain tumors. And this kills cycling cells, which means it kills your active stem cells. So if you do this to a young boy, what happens is that your dormant cells, your quiescent stem cells that are not killed by this drug, become active and regenerate the whole pool of stem cells. And this is what you see here in your equivalent 2-month-old mice. If you do the same trick when the mice is old, 22 months old, what you see is that in the same time that younger mice can regenerate the whole pool of stem cells, old mice do not. So it maybe takes longer, maybe they never achieve this. So what you see is that they are more dormant and they are more difficult to wake up. So as I told you, we found that there was interferons, when we just expose stem cells to interferons, they decrease their cycling activity, and what we did was to actually give antibodies to block one of the mediators, of the interferon response. This is CXCL10. And when we block CXCL10 in these mice, what we see is actually that we can decrease the number, these are the label-retaining cells, decrease the number of quiescent stem cells, and increase the number of active stem cells. So we have more or less a therapy, and this is you block your interferon, and then then you are well off to repair your brain. But the thing is that interferons are not all bad about interferons. Now thinking of COVID, what is interferon? Interferon is actually what is executing your antiviral response. And even more, if you think of stem cells, and this is a study of Charles Rice, interferons are there in stem cells genuinely. You don't need to activate it, but there's an intrinsic interferon response in your stem cells. And this is because we think that they are the most precious cells to protect. So this is why you have an interferon response. And they are also refractory to interferons. So you have interferons there, and they are good to fight your SARS-CoV-2. So what happens then? Should we actually block them or should we not block them? Is there a genuine function of interferons in regulated stem cell biology apart from antiviral response? And is there also in top of your intrinsic interferon response something that is constitutive? This means activated from outside, and then of course that we could block. So what we did to clarify this question was to have a look at wild-type mice and interferon knockout mice. These are interferon type 1 and type 2, so alpha and gamma receptor knockout mice that have never seen the interferon. So something that I didn't, so we did again our trick of doing single-cell sequencing. At this time, we have like 30,000 cells that we can have a look at. But one thing that you don't see in the interferon response is that it is very shallow. So you cannot catch it that good in your single-cell sequencing. So because you have to actually see what are the type 1 and then have a look at all of them together. So what we did to figure out this interferon type 1 response was to do ribosomal footprinting and to see when you expose stem cells to interferons, what are the interferon-stimulating genes that pop up in these neural stem cells. And with this, we can actually have a look at our young, this is the young stem cells, neural stem cells. So I want to make you, these are not old stem cells, these are young stem cells. And the red and yellow is the highest type 1 interferon response. Then you have non-interferon response if it is blue, and then it increases in red. And what you see here is that you have these quiescent stem cells, young, that have a high type 1 interferon response. And once they enter the differentiation, they shut down the interferon response. So this intrinsic interferon response. And then when they become neurons, the interferon response pops up again. And here are the microglia, which are the highest type 1 interferon response in our brains. And here you also have endothelial cells that also have type 1 interferon response. So this is how it looks like if you have a look at the young and the old brain now, telling you, yeah, I showed you that the interferon response is high in the young, but in the old mice, it increases a lot. This intrinsic interferon response seems to be increasing a lot. And as I said, once you enter the differentiation, then you shut down this interferon response. And increases again once you are a differentiated cell. And then you have this microglia having the highest, and then the endothelial cells. So what happens if these stem cells have never seen any interferon? So one thing you see is that your intrinsic interferon response is a bit higher in the young mouse. This means that the intrinsic is a bit counteracted by the extrinsic ligand. But indeed, you completely lose this increase induced by aging. So if you block your interferon, the aging-induced interferon response is extrinsic. This means for the pharma industry, you can block it, and actually it decreases your interferon response. And what we saw is, do these mice without interferon, how that the cell dynamic changes. And what you see here is that indeed, the mice the stem cells that didn't see any interferon don't decrease the activation rate. So they keep the same activation rate, so they are ready to go when you are old. But we don't lose them, and we don't lose them because they increase at very young ages this self-renewal. So they compensate, the system always compensates for it. And so what we have is that in interferon knockout, there's no decline of the activation rates, there's an increase of the self-renewal, and this tiny increase in the probability already has a big impact in keeping your cells. So I come to the last point of my talk, and this is the time for intervention. So what I was telling you is that, and if you have a look at the output of stem cells, this is the neurons, what you see here is that in a wild type, we lose the output. But in a knockout, we actually have a higher output at older ages. So we should actually block interferon. But I told you that if you block it too early, it seems that you actually have less progeny. So when is the right time? Now, what I'm telling you is that interferons seem to be good for the young animal, but seem to be bad in the old. So when is the time to intervene? And so what we did was to use this mathematical modeling and actually simulate at the different ages, and this is all the curves that you see here, when we would start interfering at the very young age, or at the middle, or at the end, and this is the total number of neural stem cells, but the most important thing is the output. How are our neurons doing? And what you see here is that there's a time point, like for a mouse, would be like 300 days, where it would be worth inhibiting your interferon, and then you would actually increase the output. So, I mean, this is more a philosophical take-home message. And this is like, when we think of aging and of interventions, it's not only about intervening and getting your pills as soon as possible, but it's actually thinking about, is there any good? Why does nature actually regulate these systems in this way? And what is the time to interfere with this? And this is interferon regulates stem cell output. There's a genuine stem cell function of interferons, and they are beneficial in the young, and they are actually harmful in the old. And with this, I would like to thank you for your attention, and I'm happy to address any questions.
Amazing talk. We have a question down here in front.
Great talk. I would like to come back to Parkinson's disease. So if you block interferon or CXL10 in a mouse model of Parkinson's disease when they already lost their olfaction, do you think you can restore their olfaction in the Parkinson's disease?
Well, we haven't done the experiment. I mean, what you know is that dopamine is very important to regulate proliferation of your stem cells. So this is why in Parkinson is one of the first symptoms that you have in patients. Of course, if we could, by blocking interferon, keep more stem cells, maybe, I don't know how dopamine and then interferons would actually, one would have to do the experiment. But of course, blocking inflammation in interferon, we know that you have peripheral inflammation. And on another side, another study, we know that blocking peripheral inflammation decreases progression of Parkinson.
Super interesting talk, thank you. So do you think that there are genetic variants in human population that might be affecting the interferon stem cell crosstalk? That's my first. And do you think that sex hormones have some contribution?
So I think it's a great question, because I mean we have seen, and there were many people talking about these hallmarks of aging that comes like in every talk, and something I'm always missing is the immune system, immune aging. And you know, there's more studies coming up with immune aging, and there's people that are doing this immune monitoring, and there you see that there's people that have less immune aging at the same biological age. So I'm sure that there's differences in your interferon response, and that these guys that are not immune aging are for sure having less inflammation also in the brains.
Hi. When you talk about the interferon response, you're saying these are ISGs, interferon-stimulated genes, right?
Yes.
Type 1.
Yeah.
So I was intrigued, why did you use interferon gamma receptor knockout instead of an IFNAR? Because, you know, type 1 and type 1 interferons don't bind gamma receptor.
So I actually use type 1 and type 2, alpha and gamma. So they are told.
So IFNAR is
They ignore type 1 and type 2.
You showed the data with the gamma, and I was wondering because...
No, the data was from the alpha and gamma receptor.
I see.
So they are double.
So they're double knockout. So you remove both type 1 and the gamma responses. Yeah. And when that happens, do you, at the stage when you do it, do you know that this is a response that's coming from the microglia, the type 1s or type 2 interferon being produced by the microglia that are then acting on the NSCs?
So we know, I mean, and this is also a Nature study of Wiss-Corey and Ambrunet, that there's T cells, increased amount of T cells in the aging brain, whereas type 2. And we have type 1 interferon also coming from the microglia, but we also see ligands produced by the stem cells.
All right, thank you so much, Anna. Fantastic talk. And for the speakers, Anna